Thank you very much. Lila, that was a tour de force. Really fabulous talk. So I'm really honored to be here with such distinguished colleagues that are you're making a difference for our children and our planet. So thank you very much. So I'll talk about chronic fatigue syndrome and metabolomics and then a future clinical trial. Here's my disclaimer. So the important point is in blue that Suramin has no approved uses outside of Africa and specifically in the United States. And it is illegal to import it for use in humans without FDA approval or IRB approval. So myalgic encephalomyelitis, chronic fatigue syndrome, ME/CFS, is the topic of this talk, but it turns out that we have to understand a little bit more in order to get at possible solutions. So ME/CFS is connected to these other ideas of mitochondria and the cell danger response and healing, but also includes elements of many other disorders, and I'll talk a little bit more about that. So in the United States, the National Institutes of Health is the major funder of biomedical research, and we spend a lot of time creating deep silos of knowledge. Okay, so here I've illustrated silos corresponding to our knowledge of autism, ME/CFS, Gulf War, post-traumatic stress disorder, traumatic brain injury, Lyme, pandas, suicidal ideation, other mental health disorders, aging and Alzheimer's, and all the ones in blue are ones that we've studied in clinical trials of metabolomics now. And interestingly, the insights, so although we spend so much time in biomedical research trying to identify the differences between the disorders, it turns out that they're all connected by a same root pathology. Okay. And that ultimately results in abnormal persistence of the cell danger response and specific stages of the cell danger response for different disorders. Okay, that block healing. Okay, so just in the same, if you keep opening a cut on your finger every day, it's not going to heal properly, and it'll eventually scar. Okay, so blocks in healing are what cause persistence in chronic illness. Okay, so the essential facts that I'll get across that I'll talk about today are that mitochondria have 3 developmental forms that are interconverted when a cell is injured or stressed. So I went over some of this yesterday, but for today we'll talk a little bit about. So M0 mitochondria are uncommitted mitochondria that are present in satellite cells and stem cells. Okay, these are the kinds of mitochondria you need for biomass replacement and proliferation. M1 mitochondria are pro-inflammatory, and they are specialized for defending the cell against intruders. M2 mitochondria are anti-inflammatory, and they're the ones that are really highly coupled. They run cool without a lot of heat, okay, produced by uncoupling. And those are what most people consider as the healthy mitochondria. So mitochondrial function coordinates the cell danger response, and the healing cycle is something that we've identified in molecular and chemical terms as having a 3-step sequence with a beginning, middle, and end. And that progress through the healing cycle is supported by a signaling function of the natural metabolites that are also a part of the resources that the cell uses for healing and growth. So inside the cell, those resources are building blocks that act for healing and growth. Outside the cell, they bind to receptors and change gene expression. Okay, one important thing is something that is found in all stages of the healing cycle and cell danger response is an increase in extracellular ATP signaling. So that results, that's one of the mediators of purinergic signaling. Okay, so purines are adenine-containing molecules. So, there are also purinergic receptors for pyrimidines like UDP and UDP glucose. Yeah, so blocks in the healing cycle are cause persistence of the CDR, and antipurinergic drugs or APDs include drugs like Suramin. I'm not going to tell you that we looked long and hard for across the whole modern pharmacopoeia in order to find drugs that would specifically modulate extracellular ATP signaling, and there was only one approved for human use at the time, and that was Suramin. So, an important thing, so a lot of patients spend a lot of time trying to understand the particular triggers that brought them to their chronic illness. And that leads to a certain language. And one of my messages is, let's not get hung up on terms, on semantics. So when I say the cell danger response, other people will study a component of that that's called the integrated cell stress response. Okay? And they will define that. They'll look at nucleotide metabolism. They'll look at methylation and glutathione metabolism as part of that. But that's just part of a multi-layered system of response. And in general, in chronic disease, what we find is that you cannot be sick without your gut microbiome knowing about it, and you cannot be sick without your autonomic nervous system knowing about it. And it's because they're always checking with one another. Okay, so there's a coordination between gut microbiome and the CNS and brainstem in particular. So I mentioned some of this. People will study different aspects of this and think that their particular aspect. Well, each aspect can be understood in and of itself. Okay, so whether it's innate immunity, phagocytosis, mast cell activation, the unfolded protein response, oxidative stress, oxidative burst, all these are different terms that are all part of the CDR that can be activated in a hierarchical fashion according to need. Okay, so the more severe the injury, the more systems are entrained in order to respond to that injury. So in the paper that we wrote on the healing cycle, I went through the human genome to identify G protein coupled receptors that had a DNA similarity to the receptors that would bind to extracellular ATP, and there are about 100 of them. Okay, and they actually fall into interesting functional categories that you can imagine are important early in injury. So, hemostasis and pH and lactate monitoring, Krebs cycle, leukotriene monitoring, purinergic signaling. But then products of cell membrane damage and release of sphingolipids, eicosanoids. They're also some of the viral co-receptors are actually related to our ability to respond to these interesting metabolites, these metabokines. Okay. But neuropeptides and chemokines that regulate cell chemotaxis, movement of cells into and out of the vasculature. Okay. And so this is a 7-transmembrane G protein coupled receptor that has many related. So there are about 900 different GPCRs. but these are related to the ones that respond to individual metabolites. Okay, so we know that. So in the beginning, so chronic fatigue syndrome has been around for. You can go back in the records and find things that are similar to chronic fatigue syndrome and were called post-viral myasthenia syndromes for hundreds of years in the medical literature. But it was really since the epicenter outbreak in Incline Village near Lake Tahoe in California, where there was a group of actually women's high school basketball players who came down with this devastating chronic fatigue syndrome that left them with post-exertional malaise, non-restorative sleep, episodic brain fog, autonomic abnormalities, including POTS. And then actually only a few minutes away in Truckee, there was another outbreak soon after that involving teachers at a school where, interestingly, about 30% of the cases presented with acute biliary disease. Okay, and then evolved into chronic fatigue syndrome. So we've been looking at all sorts of triggers, and so definitely there are gene triggers. There's trauma, both physical and psychological trauma play roles. EBV, Lyme, molds. There can be abnormalities in mast cells, NK cells, mitochondria, pesticide solvent, cytokines. So these are just different ways of ringing the same cellular bell, okay, that ultimately leads to a unity of response that we've called the cell danger response. I'm having trouble with this. So the topic of this conversation is going to be for ME/CFS. All right. So some of these other slides you'll have seen these from yesterday, but there'll be hopefully people from today that weren't at yesterday's talk. So. When we go from a state of health to a state of chronic illness, there are many, many paths, thousands of ways that we can become injured by physical trauma, toxins, heat. And this changes, and it's not something that is hardwired into our DNA to respond to. So it doesn't matter whether it's a cut or a scratch or a heart attack or an enterovirus infection. We have a standard way of responding to all these different things. So in the case of ME/CFS, there are a number of different causes. And the standard way is to begin the cell danger response, and there are quality control checkpoints at different stages. There's a beginning, a middle, and an end. And the important thing is that one of the things that's common to all of that as a quality control checkpoint has the extracellular nucleotide concentration, the signaling molecules that initiated the response and tell the story about cellular stress, has that been reduced over the process of healing? Okay. And so that turns out extracellular ATP is metabolized. And if it continues to be released, then the cells have not healed. They're continuously stressed. And so antipurinergic drugs can help to remodulate that. All right. So the other important thing, is that the path to illness is different than the path back to health. And so yesterday I introduced this new term. We spent a lot of time on pathogenesis and looking at pathogens that lead us to chronic illness, from pathos, for Greek for suffering, illness, or abnormality. And there are thousands of agents. But we don't have a word in the English language for tools that help to generate health-forming medications or one of those things is food, okay, but the right food. But this process has to be recognized to have a beginning and a middle and an end, and you cannot invert the sequence. Salus was a Roman god of personal health and well-being, safety and prosperity. And so we can come up with a term for salugenesis, for creating health. We can look for salugens that are molecules that help generate health as opposed to pathogens that lead to disease. And so pathogenesis, when we develop treatments to treat those acute symptoms, lead to palliative interventions. So we can, but if we actually try to promote progress through the healing cycle, then we can actually begin to see full recoveries and the improvements in resilience to future insults. Okay. So these are many ways to get to chronic fatigue syndrome, to the dark forest. We use a model in the laboratory called Dower. It's a hypometabolic syndrome that I'll talk a little bit more later. But you can get to this via mycotoxin exposure, viral exposures, Lyme, trypanosomes, trauma. I should mention at this point that it's not an accident that so sleeping sickness is caused by trypanosomiasis, but it actually is an infection-induced chronic fatigue syndrome. It's a microbial induced chronic fatigue syndrome. So these people are so fatigued, it is difficult to get out of bed. And Suramin is a drug that is used to this day to treat it. It has both antimicrobial, antiparasitic properties as well as antipurinergic properties. But these are all different triggers. I'm having trouble with the AV person. We may be losing the battery on this. But so again, the path to recovery is not the same as the path to illness, and systems biology teaches us that there are literally hundreds of abnormalities that we can identify in each person. Thank you. But these are ingress problems, and treating those don't reliably lead to a path of exit. And so I talk a lot about the difference between an engineering approach to treating acute illness, which can be highly effective and life-saving. So if a person's in diabetic ketoacidosis, insulin and fluids can be life-saving. But insulin never cured diabetes. Okay, it's a deeper problem, and so we need to have a biological approach to recovery. So pathogenesis versus salugenesis. Salugenesis leading to recoveries. So this is a fun slide that is a mustard plant, a Arabidopsis, that has been engineered to fluoresce when intracellular calcium is reduced. Okay, and let's try this again. Okay, so yesterday this animation worked. Today it doesn't work. So what this would show is that thank you. Okay, so over here, there's a little cricket bite in the leaf, and the signaling molecules that are used are either glutamate or ATP, which are highly concentrated within the cells, and when cells perceive that release of extracellular glutamate or ATP, it propagates the signal of danger to the whole rest of the plant within 2 minutes. Oh yeah, could you show the video one more time? So, yeah, it's exciting every time. So, okay, so, all right. So, so this is an ancient response. It doesn't matter, if we're talking plants or animals, but the fully formed cell danger response requires multicellular organisms. There are elements of this that can be studied in single cell organisms as well. But not the fully formed story. So during health, we cycle between wakeful activity and nutrient intake and restorative sleep. And that's the normal cycle that we live in. But new information and learning and stress and development, pollution, all of physical injuries, all of ways of introducing stress, and we can, if we have more severe types of stress, there's more release of extracellular ATP and other metabolites, okay, not just ATP, from specifically evolved channels in the membrane that are designed to actually help facilitate cell-to-cell communication. Okay, and so when a cell is infected by a virus, it releases more ATP to warn its neighbors that there's danger in the neighborhood. Okay, so the first stage is containment, and so what can happen in this stage is not unlike what happens when a submarine, a section of a submarine has a breach in the hull. The compartment doors get closed in order to contain the injury. Okay, and there is a move toward cell autonomous control and a breakage away from systems control from autonomic and neuroendocrine pathways, and cells become, in this first stage, cells do what nations do when they go to war, is they harden their membranes and they don't exchange resources with their neighbors. Because if you're a cell under attack, a neighbor could have a virus. Okay, and so until that's contained, communication between cells is reduced. Okay, so once the beginning, the middle, and the end of the cycle has been completed, then the extracellular ATP levels are reduced, as well as other signaling molecules. But that reduction allows you to return to the health development cycle. Okay. Each of these stages has a measurable and characteristic metabolism. Okay. These are things that actually have been known for many years. That the early stage of inflammation requires glycolytic metabolism. In order to proliferate, to replace the cells that have been lost, it requires aerobic glycolysis. And in order to redifferentiate and to remodel gene expression to meet the needs of the differentiated cell, we have to have oxidative phosphorylation. And then once we return to full health, both OXPHOS and glycolysis and aerobic glycolysis are used in a blended fashion and alter in a circadian way in response to food and activity, and are interconnected and coordinated by both the autonomic nervous system and the gut microbiome. Okay. So we look for different stages of blocks, and autism and ME/CFS appear to have blocks at the CD3 checkpoint 3. This is an expanded version of this. This hasn't been published yet, but it's where we've actually identified substages within each stage of the CDR that have slightly different characteristics. So within the beginning, the middle, and the end, there's an early and a late stage to each. Okay, and in chronic fatigue syndrome, one of the things that we've seen is that during this phenomenon called crashes, okay, where there's a post-exertional malaise, that post-exertional malaise is associated with an inability to, the patients who experience this have moved into a post-lactate threshold phase of exercise performance. We can induce this on an exercise treadmill or a bicycle. A normal person will be able to recover within an hour to two. A patient with chronic fatigue syndrome can take several days before that post-lactate threshold has been recovered to normal metabolism again. And during that time, they will be unable to generate the same amount of activity, and will have to take time off. And how that manifests, many of you know. So a patient may be able to go do their own grocery shopping in a day, and they might have 2 bags, but they'll bring them home and put them on the counter and wait until the next day to put the groceries away, because they've exhausted their energy, their activity capacity for that day. Okay. All right. So this is a picture of a water balloon that has thousands of little holes in it. Okay. And the more pressure you put on the water balloon, the more little fountains of water come out. And you can think of ATP as the life's blood of the cell, the energetic life's blood of the cell. And the more stressed a cell is, the composition of the plasma membrane, the lipids around the channels that are made up of pannexin and P2X7 and connexin channels will actually pull apart. This also happens as the redox potential of the cell becomes more oxidizing, and more disulfide bonds begin to form from free thiols. And so those disulfide bonds also open up this channel and increase the conductance to the outside. And so the concentration of ATP is about 1,000 times more than immediately outside. So inside it's about 5 millimolar. Outside in this pericellular zone of unstirred water, it's about 5 micromolar. Okay, so all stress cells leak ATP. That's how the cell tells its neighbors that it's under physiologic stress. So mitochondria will sense this. They will talk to the nucleus that results in new changes in gene expression. Remember, all but 13 proteins in mitochondria are made by the nucleus, and so there's about a 1,500 protein proteome of mitochondria that differs according to different cell types. Every different cell type in the body has a different mitochondrial proteome, but only 13 of those proteins are made by the mitochondrial DNA itself, and all the others are made by gene expression from the nucleus according to the needs of the cell. Okay, so the cell will begin to adapt mitochondria to meet the stress that is signaled both in a cell, an autocrine fashion. So ATP will, so we call this short path retrograde cycle to the nucleus, and then the long path retrograde signal is from molecules released by the cell that then bind to the cell as well as neighboring cells to change gene expression to better facilitate the response to to stress of any different kind. So with all these channels in the membrane, if a cell is losing a significant proportion of the ATP that it's spent making, then it's harder to maintain adequate amounts inside the cell. Okay? And so we went looking for a drug that might be able to block that loss of extracellular ATP, and as well as to modulate the signal in a way that decreased the cell danger response. Okay. So how does this relate to chronic fatigue syndrome? It turns out that all life on the planet has developed many different ways to respond to environmental stress in a hypometabolic conservation way. So there are persister cells in Lyme, TB, many other bacteria. After fertilization, many animals will actually implant, but arrest fetal embryo development for many months, where there's a kind of cessation of biological time, even though chronological time continues. So mice and humans are among only 2% of mammals that don't have this phenomenon. Many, so for example, bears will mate in the fall, implant, and then there'll be an arrest of the embryo until a period farther along through the winter during hibernation, and then we'll begin growth again. So there are specific controls. Hibernation is another one. Torpor is a hypometabolic response that hummingbirds will use to get through the night. Estivation is a hypometabolic response to high temperatures that many reptiles will have. Tune is a cryptobiotic form of the tardigrade, the little water bear, that allows it to actually survive radiation from in space to reduce its water content to barely detectable, but then can reanimate when the environment becomes safe again. And Dauer is the phenomenon, a German word for persistence, or to last, that we study in the laboratory with a little nematode classic genetic model called C. elegans, Caenorhabditis elegans. Okay. All right, caloric restriction and longevity. So also will induce a reduced metabolic, basal metabolic rate, okay, that is also energy conserving and will result in improved mitochondrial quality control. Let me put it that way. Okay. Even plants will arrest their embryos in seeds, and they will, just before entering into seed production, they will reallocate energy for making oils. And the oils for the seeds act as a food source for germination and growth of the embryo. But it's kind of like an egg. The fuel source is also the yolk, okay, that then allows for recovery of metabolism once the environment is safe again. Okay. All right, so we're interested in this molecule of ATP and its signaling, and specifically extracellular ATP. We're specifically wanting to explore the therapeutic efficacy of an approach to treating chronic illness that's called molecular armistice therapy, where we try to use signaling molecules to send the message that the war is over. Okay. Now, all you may know in a toxic environment, not all battles of the war have been fought. Okay, but what we've learned is you don't have to fight all the time. Okay, so this is the drug Suramin. The sulfonate groups, these 6 sulfonate groups, will mimic the negative charges on the phosphates of ATP. It's 126 atoms of elements that all have to be put together in the right way in order to do this. That's important because this was a molecule that was actually synthesized in 1916 on the heels of Paul Ehrlich's work to try to find cures for, initially syphilis, but then African trypanosomiasis. So what we're really looking at is how the cell danger response can block healing, and how antipurinergic therapy can lead to improvements. Okay, so this is an animate. Let me see if this animates. Okay, no. So there's an animation associated with this that you might be able to press. Okay, so in a small phase 1/2 clinical trial, we looked at a single dose of Suramin in 5 boys. And Suramin's the 100-year-old drug that was called Bayer 205 to test the idea that ASD could be treatable, a treatable metabolic problem. So here's a neuron, a nucleus, and mitochondria. There are pores in the membrane that allow for ATP to be released. And there are also cell surface receptors that respond to ATP. And so a stress cell leaks that ATP, and that ATP binds to the receptors, and it changes the gene expression pattern of that cell. Okay. Now, if we treat with Suramin, it can block the loss of ATP from the cell and compete with ATP for binding at the receptors, so that energy that was being allocated for defense can now be reallocated for growth and development. All right. Okay, so I'll tell you briefly about the first Suramin autism treatment trial 1. And the main thing that I want to get across is that autism is defined by an ADOS score of 7 to 10, so that's autism spectrum is a severity of 7 to 10 is the highest level. Our average child had 8.6, and after a single dose in 6 weeks, they had dropped to an average score of 7, right on the threshold of coming off spectrum. Okay, and the question that we have, and there were no significant differences. You can see the error bars are equivalent here in the placebo group. The question that we have is, if we give 3 doses, can some children come off spectrum? Okay. And so we did something that had not been done in 100 years of using Suramin, is we tried a dose that was not designed to be antimicrobial. It was not designed to be chemotherapeutic for cancer. Okay, it was a low dose that specifically produced blood levels of 5 to 15 micromolar. Okay, and we saw no significant safety impacts compared to placebo in this study. But in sleeping sickness, the blood levels are about 50 to 100 micromolar, and they get about a month. But because of the long half-life of the drug, they can maintain antimicrobial concentrations in the blood for 4 to 6 months, even though the treatment only lasts a month. And in cancer chemotherapies, that the levels that were increased, say if this is an average of 10 to an average of 250, so 25 times higher that you need to achieve to kill cells. So that was the purpose of using high-dose Suramin in cancer chemotherapy studies. But as you might think, higher doses that are designed to produce cytotoxicity come with a risk of side effects. And under these chemotherapy doses, about 10% of patients per month would develop side effects, the most common being an adrenal insufficiency, but anemias and peripheral neuropathies were also seen. But this is, surprisingly, Suramin has actually a wider therapeutic index than aspirin. So if you were to take 2 aspirin for a headache, you might say that's safe and effective. If you were to take 50, 25 times more, same amount more, okay, that would land you in a coma or worse, okay? So dose, the dose matters. Okay, so I'm going to move through the metabolomics end of things a little bit quickly, just to point out that we can use the blood to identify similarities between many apparently different kinds of diseases. And that when this is done, the diagnostic utility of this can be very high in autism and PTSD and Gulf War illness, chronic fatigue, and major depressive disorder. There'll be papers that will be coming out this year that will be in Gulf War illness. We're reporting on this, on major depressive disorder with suicidal ideation, and on recurrent major depressive disorder. So once you have an episode of clinical depression, the risk of subsequent episodes is 6-fold greater than before. We can identify that in the signature in the chemistry. But anyway, all these different diseases are allowing us to see the commonalities between them. So not just the silos that are independent, but also the roots that are the same. So we published a paper on the metabolomic features of chronic fatigue syndrome in the summer of 2016, and within just a couple weeks, this cartoon appeared, by a talented artist, S. Jones. I don't know this person, but it was cute, and it basically pointed out that NIH had been hibernating on the topic of chronic fatigue syndrome for about 30 years, and that when we can identify a signature in the blood, it gives us a tool to move forward, and now we can come out and get to work. All right, so studies have been done by others besides us that have looked at so many of these different disorders are pathway disturbances in chronic fatigue syndrome have now been replicated. So Maureen Hansen just published a paper last month where 98% of the pathway abnormalities that we found, she replicated in her study. And when studies are done, like what happens after, let's say, a Zostavax vaccination, and metabolomic studies have been published by, say, Lee and Cell in 2017 that looked at how a vaccination actually interestingly produces changes in sphingolipids, sterols, purines, methionine, cysteine, propionate, inositol lipids, porphyrin and heme. So many of these are shared. Turns out that 85% or 12 of 14 different pathways that were found in a post-vaccination metabolomic study are also seen as disturbed in chronic fatigue syndrome. Okay, so we've been in a holding pattern for pushing beyond 3 years now. Because of a sad but true fact of the history of Suramin, which is that it's 100 years out of patent, which makes having a sponsor willing to put in the effort to develop the clinical trials. Well, it makes it hard for for them to have the profit margin that they want. Okay, so in autism, I can the amount of drug that we need, with this low dose regimen, is I can treat an 8-year-old for a year for $27 worth of Suramin. Okay, so that does not incentivize. So and to give you a feel for what people need for what the Food and Drug Administration needs to approve a new use of drug is you need to go through a set of clinical trials ending in a multi-center phase 3 clinical trial that typically for autism will range, it'll be about up to $50 million. I'll just say that, $50 million. And that's not money that's easy to raise from grassroots. It really takes, profit incentive, in order to come up with that. And it's only been relatively recently that we were able to get a sponsor that's created a little biotech startup to take on the job of making new Suramin and supporting all of the clinical trials necessary that the FDA needed to determine the safety and efficacy and approve the drug. I have an approved FDA investigational new drug application we've had for 3 years, okay, to do this initial pilot study, as well as our institutional review board approval. But it's a randomized, double-blind, placebo-controlled trial of 10 females with ME/CFS that will be matched and according to age, severity, and duration of illness, and then randomized to either placebo or Suramin. They'll get 2 doses in 2 months, and then we'll look at how they did after 6 weeks. Cost of that's about $900,000. Okay, so this is the way it looks. We've got about 10 pairs of participants that are matched according to their Karnofsky score, the duration and severity of ME/CFS. And they'll get put randomly into Arm A and Arm B. And they'll receive a dose at 1 month, or a dose at 0 time and a dose at 1 month, and then we'll evaluate daily activity. We'll use the gold standard for total energy expenditure, which is called the doubly labeled water technique that uses deuterated water and H2O18 in order to calculate daily caloric utilization. And we'll look at sleep and many other parameters. So the idea is, so the question is, can people begin to return to a road to recovery if they receive some Suramin? So that's that. Primary outcomes: safety, Karnofsky score, and the total energy expenditure by doubly labeled water. We have secondary outcomes, including the SF-36, but a lot of other things. I think that's it. So if you want to read more about the metabolic features of the cell cycle, antipurinergic therapy, and Suramin, we have some references, and you can get a lot of the references actually tied to our webpage. And other than that, I'll say thank you very much.
Thanks very much, Bob.
Sure.
It's absolutely fantastic. We're just going to have a little bit of a slight interim before going on to the questions. We'll get Lila back up on stage, just because one of the people we're going to speak has only a limited opportunity. It's just to talk about, so we'll get Bob and Lila back up on stage.
So there's more time.
So there's more time to talk.